guys, in this video we're going to talk about a variety of viruses. We're going to talk about the Poxviridae. This is basically the family of viruses that causes pox. The most famous and most notable of these viruses is smallpox, which thankfully has been eradicated. But we still teach it. And we still teach it because if we see it again, it is unfortunately likely to be a mechanism of war. So you need to be able to recognize it and understand what it is and know what the appropriate treatment options are. There is a vaccine for it, which is used in some individuals, and we'll go through that. The vaccine is actually built on the vaccinia backbone, which is another pox virus that really is only used to create the smallpox vaccine. Then we're going to talk about all of these guys: orf, cowpox, monkeypox, and Molluscum contagiosum, which are actually pox viruses that we do still see, so you may want to be aware of their presentation. Okay, so let's just go kind of the main characteristics for all pox viruses. The Poxviridae are actually the largest, most complex viruses. They have a linear double-stranded DNA genome with kind of fused ends. This is a DNA virus which replicates in the cytoplasm, which, as you know, is very strange. Normally, DNA viruses replicate in the nucleus, but not these guys. They replicate in the cytoplasm, which actually leads to the formation of inclusion bodies, which I'm actually showing here. The inclusion bodies are known as guanary bodies, and here's a really bad microbiology joke for you. You can associate guanary bodies with smallpox because smallpox is all guan. Yay. Okay, bad joke. But it does help you remember it. Trust me, one day you're going to be taking an exam and you're going to chuckle to yourself saying, that Dr. Richards, her dad jokes pay off sometimes. Anyway, it encodes and carries all proteins necessary for mRNA synthesis, which makes sense. It's a huge virus, so it better actually have some luggage in that virion that actually allows it to replicate itself. So this is kind of some pictorial, but you can actually see, look at how massive, even on EM, this virus is, and the giant inclusion bodies that we can see. And the inclusion bodies are actually kind of little factories, little machines of virion production. All right, so we'll talk through the structure and replication of this. The replication of pox viruses is unique among all the DNA-containing viruses in that the entire multiplication cycle takes place in the cytoplasm, which is very, very strange for a DNA virus. Therefore, the pox viruses have to encode everything they need. So they need an enzyme for mRNA and DNA synthesis because they won't be borrowing these from the host cell because they never actually go into the nucleus where those could be found. And they also have to include anything else they might need that a DNA virus would normally obtain from the host cell within the nucleus because they are never going to go in there. So we can just kind of forget that this cell even has one. So how does it work? So first off, the pox virus, like any other virus, is going to bind to its cell surface receptor on the outer membrane, and it's going to fuse, okay, with the cellular membranes, either at the cell surface or within the cell. The core is basically going to be liberated using an enzyme known as uncoatase. So it's going to be uncoated using the uncoatase enzyme. This removes the core membrane, liberating the viral DNA into the cell cytoplasm. At that point, you're going to have early enzymes like uncoatase that created this uncoating. And at this point, you're going to have early gene transcription, okay? The early gene transcription basically contains a specific transcriptional activator and all the enzymes necessary for transcription, which would include the RNA and DNA polymerases, basically. So the viral DNA replicates in an electron-dense cytoplasmic inclusion, which are the guanary bodies, okay, which I showed you on the previous slide. And like I said, those are just little factories. Later, you're going to get late viral mRNA for structural virion and other proteins that are produced after DNA replication. So we do this DNA replication. It takes about 2 to 5 hours. Now we begin late mRNA, late transcription, and we use the late enzymes to basically create these late virion proteins. All right, so you have early virion proteins and late virion proteins. And altogether, these are going to take place around the guanary inclusion body. The membranes are going to assemble um, around the core factories, and approximately 10,000 viral particles are produced per infected cell, and different forms of viruses are released by exocytosis or upon cell lysis. But it really doesn't matter how the virus gets out of the cell, because no matter how it does it, the virion is infectious. So this is a complex and smart virus that creates a very specific cytoplasmic or cytopathic effect, the guanary body, and it's a DNA virus that replicates in the cytoplasm, which is just super weird. So I should mention that all of the steps that I just mentioned in replication are true for all of the pox viruses, except for Molluscum contagiosum. Molluscum contagiosum is restricted to keratinocytes. Um, and it's basically going to stimulate the growth of the cell and prevent apoptosis, inhibiting inflammation, and it's non-cytolytic. In this way, it actually acts a lot like the human papillomavirus, HPV, which is released when keratinocytes mature and senesce. So Molluscum contagiosum really is non-cytolytic, whereas all of the other smallpox viruses, they can be either cytolytic or they can just kind of leave the cell and either way produce infectious virions. All right, so I'm going to go through kind of the pathogenesis and immunity of smallpox, and then I'll go through it for Molluscum contagiosum. And for Molluscum contagiosum, you can actually apply that to all of the other pox viruses that we're going to talk about. But first, let's talk about smallpox. Smallpox is actually an inhaled respiratory virus. So you initially inhale it. This is another one of those viruses that when it used to spread, it was often spread on textiles, blankets, things like that, where the virus was actually in them. People shake them to clean them, etc., and then they could breathe it in. And then obviously spread when people cough, through droplets, aerosols, etc. So anytime something is airborne, it's obviously a significant concern. So even though technically it's eradicated right now, any live viral stocks of smallpox could theoretically become a bioterrorism weapon. So dissemination of the virus from the upper respiratory tract occurs once it hits basically the lymphatics. So you get infection of the lymphocytes, specifically macrophages, and that leads to a cell-associated viremic spread through the lymph node and into the blood. Eventually, you're going to get infection of internal and dermal tissues, which are inoculated after you get your secondary viremia. And it's this secondary viremia that eventually leads to the characteristic pox-like rash. The initial viremia actually leads to infection of the spleen, the liver, the bone marrow, and any additional lymph nodes and organs. But it's the secondary viremia that actually leads to the characteristic pox and rash that we expect to see. All right, Molluscum contagiosum. This one is not respiratory transmitted. It's actually transmitted through direct contact with lesions. And that's actually true for horse and monkeypox and cowpox largely as well. In this case, you're going to see a wart-like lesion. And for all of these viruses, cell-mediated immunity is really your only way to kind of survive this. It's essential for resolving a pox virus. And what's really tricky about that is up to 30% of the genome is actually designed to inhibit the cell-mediated immune response. So it's a smart little virus, and I shouldn't say little because, like I said, it's a pretty big virus, but, you know, comparatively to us. But this bugger is really adept at fighting us. So they have genes that basically are devoted to activities that help the virus evade immune control, which include proteins like impeding interferon, complement, inflammatory effects, antibody, and cell-mediated protective immune response. So just to recap, it can inhibit the innate immune response through interferon and complement inhibition, and the humoral immune response through antibody inhibition, and the cell-mediated immune response through basically inhibiting the effect of cell-mediated immunity. So with all of that, it's no wonder that it can very quickly kind of spread if you do not have a good cell-mediated immune response. So talking about the epidemiology of an extinct virus is a little strange, but we're going to do it anyway. When there was variola, which I don't think I've said that yet, but variola virus is actually the name of the smallpox virus. We were the only source, humans. And that's actually part of why we were able to eradicate it, because it was just us. So if we could just vaccinate all of us and make sure that nobody else got it, we actually were able to get rid of it. And it has at this time been eradicated. There are some, live stocks of it, I'm sure, in some laboratory somewhere, but it is not currently circulating in the human population. When it is transmitted by aerosols or contact with lesion material or by fomite. When we talk about Molluscum contagiosum virus, this one is actually spread by contact. So it can be spread through actually just, touching a lesion, sexual contact. And then we also associate it with wrestlers. Wrestlers actually tend to have a higher incidence of Molluscum contagiosum because they can actually get it in two ways: direct contact through their wrestling partner or also by fomites. All of the other pox viruses, vaccinia, orf, cowpox, monkeypox, are zoonoses. They infect humans and other species, and they only tend to infect humans through accidental or occupational exposure. So you can see there's certain places where we expect to see them. So orf you get from sheep and goats, cowpox, rodents, cats, cows, and monkeypox, monkeys and squirrels. So as I mentioned in the rabies video, squirrels will not give you rabies, but they might give you monkeypox. The other thing that I just want to touch on with smallpox that I don't know if I've kind of brought it up, when it was, circulating, it was a highly, highly contagious virus. And it's so contagious that there actually is a vaccine effect because it is a live vaccine built on the vaccinia backbone. So at this point, we really only vaccinate military personnel, and that has brought the incidence of vaccine-mediated disease in contacts down. And there really isn't need to vaccinate most of the population. I know it's strange. I'm telling you not to get a vaccine, but really for the safety of the population, it's better that we only vaccinate people who have a higher risk of potentially coming in contact with it. All right, let's talk a little bit more about smallpox. There are were two major variants of smallpox. Variola major, which had a really high mortality rate, 15 to 40%, and then variola minor, which had a mortality rate of only 1%, but was still a fairly significant disease. There's a 5 to 17-day incubation period in where the infected person will then begin to experience a normal viral prodrome. That viral prodrome will include things like fever and fatigue and malaise, or I guess that's fatigue and myalgia, the normal things. They may also experience a severe headache, backache, etc. They then get this vesicular rash. You can see it in the mouth, although I don't think I'm showing that here, and soon after on the body. It's not uncommon for patients to vomit, have diarrhea, and then eventually excessive bleeding. You basically get a simultaneous outbreak of the vesicular rash kind of all over the body. You can see it here on the trunks, on the face, on the soles of this baby's feet, on the hands. And basically, because it's a simultaneous outbreak across the entire body, that's one way to distinguish smallpox from the vesicles of varicella zoster, because zoster actually erupts in kind of successive crops, right? Whereas this is just whoosh, all of it at once, okay? All right, so we stopped it. How? The eradication of smallpox is one of the greatest triumphs of medical epidemiology, microbiology, immunology. It's the most amazing thing we've been able to do, I think, thus far, because this was a killer and it was massive. It was due to a massive World Health campaign to vaccinate all susceptible people, especially those exposed to anyone in disease. And it was actually declared eradicated in 1980, so not even all that long ago. So how did we control it? Well, the early approach to controlling it was the concept of variolation. Variolation was basically an early immunization. Immunization, basically, when you're talking about variolation, what they did was they would take one of the pox and they would pull some of the virulent smallpox pus out of the pox and then basically create a scratch on an uninfected person and allow them to get the disease. The thought being that it would be a lower or less virulent strain or a less severe disease. It was used in the Far East and later in England. Unfortunately, though, it didn't always work out that way. The fatality rate of variolation was about 1%, the same as variola minor. And there's a lot of reasons for that. Partially, we can't really control the dose in that case. We can't control how severe the disease would be, and you also can't control somebody's immune response. So I think the goal most of the time was to take somebody who was having kind of a less severe form of smallpox and use them to variolate another person. But if they have a really low titer of the virus, let's say, because their body was really well controlling it, you might not give enough to create a protective response in the other person. Or let's say they just had a really great immune response, so their response made it so the disease didn't seem that pathogenic, but then you put it in someone who doesn't have as good an immune response and it's super pathogenic, and that person gets a really severe disease. So for a lot of reasons, variolation, I understand why people did it, but it wasn't ideal. So then, thankfully, we moved on to vaccines. So the vaccines are basically built on the vaccinia virus. Okay, so we're using the vaccinia virus to create this. The vaccinia virus is actually a hybrid of pox viruses, and there are actually 3 smallpox vaccines. There's the ACAM 2000. This is the only licensed smallpox vaccine in the United States. It is a replication competent, i.e. live, vaccine, which provides for active immunization against smallpox disease in persons determined to be at high risk for smallpox infection. The vaccine does not actually contain the variola virus, and therefore it cannot cause smallpox. It's not the same virus. Variola and vaccinia share a lot of things in common, so the immune response to one can be protective to the other, but it is not the same virus. It contains vaccinia virus. So if you get the smallpox vaccine, you have not actually been exposed to smallpox, but you are protected against it. The second vaccine is the Aventis Pasteur smallpox vaccine. This is also a replication competent investigational vaccine. It may be used in a smallpox emergency. And then there's Imvamune. Imvamune is a non-replication competent vaccine. So it's dead or subunit. It's also an investigational vaccine which may be used in a smallpox emergency under the appropriate regulatory mechanism. Now, suddenly I'm speaking like a government robot. Why? Because I basically lifted that directly off of the CDC site on immune vaccination of smallpox. So basically, there are stockpiles of these vaccines which will be used at the discretion, and they claim that they have enough for everyone. So I'm sleeping well at night. All right, so vaccination procedure. The vaccine procedure consists of basically scratching the live virus into a patient's skin, okay? So you're basically going to take this bifurcated needle, and you can kind of see here where the needle hit both spots, and put some of the replication competent ACAM 2000 into the patient's skin. You're then going to look for what's called a take. A take is a pustule. If the pustule doesn't develop, then the patient didn't actually get enough of the virus in order to actually be protected. So you can see you're waiting for kind of this giant pustule to form and then to go down in size, okay? As the incidence of the smallpox virus waned, the risk of smallpox development was low, but the risk of complications related to receiving the vaccinia vaccine related disease actually rose. So this is what led to the discontinuation of routine smallpox vaccination and is now largely only done for military personnel and first responders in case of biowarfare. So let's talk about a couple of the complications. If you were to receive the vaccinia vaccine, there is the potential to have specific complications because it's a replication competent virus. So this would include things like encephalitis and progressive infection, which is known as vaccinia necrosum. And this is most likely to occur in an immunocompromised patient that were inadvertently vaccinated or made contact with somebody that was vaccinated. And patients in this case might receive vaccinia immunoglobulin and antiviral drugs to help protect them. All right, that is enough on smallpox. We're now going to talk about Molluscum contagiosum. Molluscum contagiosum is actually a really common disease. It affects about 3 to 20% of the population. The lesions of Molluscum contagiosum actually differ from other pox-like lesions because they're kind of nodular and wart-like. You can see them here. They begin as papules, and then they become kind of pearl umbilicated nodules that have a central caseous plug that can be squeezed out. They're most common on the trunk and genitalia and proximal extremities, and usually occur in a cluster of about 5 to 20 nodules. So you can see a nice cluster here. There's lots of little nodules there on that individual. The incubation period for Molluscum contagiosum is anywhere from 2 to 8 weeks. The nodules are typically going to resolve on their own, but it's going to take time, 6 to 12 months, and in some cases as long as about 4 years. The disease is much more common in children than adults, and also in wrestlers. This is one of those ones that we see in them. But its incidence is increasing in sexually active and immunocompromised individuals, so something to look out for there. Okay, orth, cowpox, and monkeypox. These are also pox-like viruses, or they are pox viruses. Human infection with orth and cowpox is typically an occupational hazard, resulting from direct contact with the lesions on the animals, so farmers. A single nodular lesion will typically form on the point of contact, like the finger. So in this case, basically what you would see is either a hemorrhagic or a granulomatous lesion. So this is actually a granulomatous lesion. It's caused by orth. If you saw a hemorrhagic lesion, that's more likely cowpox. The lesion frequently develops and then regresses in a little over a month, 25 to 35 days, and typically no scar forms. The virus can be grown in culture or seen directly on EM, but it's normally just diagnosed from symptoms and the patient's history. Monkeypox is kind of like a milder smallpox. You're going to get that pock-like rash. I'm showing kind of small images of it here. But it's not as severe a disease as smallpox. In the case of this one, you're going to have a pock-like rash that happens after a viral prodromal period. The viral prodrome period is about 1 to 3 days. The incubation is 7 to 21 days after exposure. During the prodrome, patient will have a fever, might develop the rash. It normally begins on the face and then spreads to other parts of the bodies. The lesions actually progress through stages, from macules to papules to vesicles to pustules to scabs before just falling off. Typically, the illness takes about 2 to 4 weeks to clear their system. We don't see this one a lot, but as luck would have it, in about 2003, Illinois actually had an outbreak of monkeypox. There was actually 100 cases of the illness of monkeypox seen in kind of the Midwest area: Missouri, Wisconsin, Indiana, Illinois, and then Kansas. This actually happened when prairie dogs were purchased as pets. So basically, these prairie dogs had been housed with Gambian giant rats, which apparently were infected with monkeypox. People bought the prairie dogs, and then they got sick with monkeypox. So this is kind of an odd way of saying that even though we don't see it a lot, theoretically, it could happen.